Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola, ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us, the 139th seminar, and I call it a Max Planck Institute of Biochemistry Day. I got back from my 2-week trip to the UK and Germany. It's coincidence because we have two speakers from both countries today. And then I just came back to give 5 invited talks at Imperial, UCL, and Cambridge, but not Oxford. I'm sorry, as well as 2 conferences covering synthetic biology and nucleic acid. And unfortunately, I was also nearby, the Petra, today's pioneers, place, the Munich, but that's not the location. It has been an amazing trip to meet a lot of great people and young researchers to discuss current and future projects, and actually I form a collaboration in UK professor. I will embark on another domestic and international trip for 2 weeks to San Francisco or a DOE, Department of Energy JGI meeting in Korea, now in Asia. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Petra Schwille. I hope I pronounced her last name correctly. She is the director of cellular and molecular biophysics at Max Planck Institute of Biochemistry, Germany. And that's why I call today is Max Planck Institute of Biochemistry Day. I recently met her at a conference, but her perspective paper titled Bottom-up Synthetic Biology, Engineering in a Tinkerer's World, which was published in Science 13 years ago, basically inspired me to summarize my views on synthetic biology just before I applied for faculty jobs. I even used the genetic part box figure in that paper for my introduction slide for my talk many times. So I wanted to thank again her one more time for her amazing contribution to the scientific society by sharing her vision and helping me personally shape my vision 13 years ago with the amazing perspective article she published many years ago. Petra, thanks again, and the virtual podium is all yours now, and thank you.
Yeah, thank you very much. This is a very kind introduction, and it's also a nice opportunity to be a supporting act in this case here, and I try to go not too much over time, giving a brief introduction into what our research rationale is. You referred to our little graph of the building system. This is basically the 2.0 version of it, which is not even by ourselves. But the idea is really to take the engineers or the bottom-up approach to cellular functionality in general and to the total of a cell in particular. I mean, we still don't know what a living system really needs to have. What are the defining features that need to be in every single cell? And we can only find out, I believe, if we try to build it. And how do we build it? I mean, there are, of course, many ways to approach the system. Ways more from chemistry, which are focused very much on the generation of the first biomolecules, the so-called origin of life field. that our the other speaker has connections to. My particular angle to the question is more a modular understanding. So not so much the very molecules, the very chemistry, but more the functionality. Is it possible to dissect a living system or parts of a living systems into very few modules, if you bring them together from scratch, that they would show this functionality? And in a way, it is synthetic, of course, in nature, because you bring things together, but it has, of course, also a lot of overlap with synthetic biology in thinking about living system generally as being modular. Now, what modules are we taking? We are taking proteins, because functionalities are best represented so far with proteins, knowing, of course, that proteins are the end point of evolution in a way, right? And that was probably not the material that the first cells used to engineer their functions. So how are we approaching it? We basically, for some reason I cannot change. Here. We are engineering, so we are very much concerned with the compartment vision, that a cell needs to be a compartment of aqueous, aqueous molecules. And what kind of a compartment would make sense? Of course, a compartment that is similar in the building blocks to cells, which is phospholipid membranes. So what we're building technology-wise is making vesicles, large ones, about the size of cells, so that we can study them with advanced microscopy and introduce into these vesicles our functional modules. And this introduction is sometimes easy, sometimes not so easy. Of course, you have to standardize. You have to make sure that you make all your modules happy. Which is not entirely easy. All you know is that cells are a huge mixture of many different proteins, and some functions that we understand in living cells might only unfold in this complex environment. And we all know this, all protein reconstituting protein biochemists people know, of course, that it is very likely that you observe a fundamental function, a very obvious function in a real cell, and as soon as you want to take the protein out of the cell, that was it. Well, of course that happens, but of course we're trying to get hand of proteins that do function in our simplified environment. We have recently, together with other people in the field, come up with a lot of different methods to make these vesicles, to fill them with protein solution, buffer solution. Microfluidics is very prominent for that. What you've seen here is a microfluidic production of these vesicles, which is superior in the way that you have a very good size distribution. That's all, can be reproducible in size and content. What is our latest news? Probably not so latest anymore. It has been published quite some time ago, but just to give you in a nutshell, what are the greatest breakthroughs that this approach has yielded in our hands was that we basically go from a very important feature of life, namely the ability to spatiotemporally self-organize upon energy dissipation. That's basically fundamental for all living systems, to one, I would say, very important feature of life, especially for physicists, which is cell division. Why for physicists? Because it's a mechanical process. It's something that you can easily watch under your microscope, and you can easily see whether it happens or not. And it has to do, of course, with the introduction of mechanical forces by these functional elements to the compartment that they are in. So in principle, you can ask the question, what kind of molecules do I have to encapsulate into a vesicle system that are able to, upon energy supply, ATP or any kind of energy, self-organize and autonomously divide the compartment that they are in. So that's the technical question, if you will. And we've addressed that with a very beautiful set of proteins that we've taken from E. coli. which are, first of all, the so-called Min proteins, which regulate the positioning of the division axis in E. coli. This is a video showing that these proteins carry out pole-to-pole oscillations in the cell, and these pole-to-pole oscillations are supposed to position the division ring to the middle. We've basically reconstituted these proteins, and here I probably should say there's the modeling of this process. There are two proteins together. They organize into the in these traveling or oscillating waves. That was known. What we've done is we basically reconstituted them in vitro, first on planar membranes to show that they're actually able to produce these beautiful patterns also in much simplified settings. And lately also in closed compartments. If you shape your closed compartments that they look like bacterial cells, you can appreciate that the pole-to-pole oscillations, although in here we have only the two protein kinds and the ATP and the membrane that they bind to, is very nicely reproduced. And lately, we've been able to bring all this into a freestanding vesicle, basically a vesicle that is no longer somehow, here it was a technical volume microstructure that we lined with membrane. Here we really have freestanding vesicles that are filled with in vitro expressing machinery that makes 5 proteins altogether, 2 proteins that are basically responsible for the oscillations, and 2 proteins that are responsible for building something like a division ring. And here you can see very beautifully that over time, first the network of a cytoskeleton is formed, and then once the oscillations start, this network is basically pushed together in the middle at the geometric center or at the equator of the vesicle. It runs again. As you can see how it basically autonomously transfers from a relatively isotropic cytoskeletal network to, upon the self-organization of the oscillation, to a more or less defined ring structure that, and that's very important, induces the first mechanical change to the vesicle. We are basically here in this picture down here. So we now have to find out what next steps we need to make this fission complete. Engineering proteins, engineering the size. We'll see what we need to do, but that's where we are at the moment. And with this, I would like to stop. And thank you very much for your attention, and hand over to the main speaker for today. And I'm very much looking forward to your talk.
I have a quick question before.
Oh,
Sure.
of course, of
It's
course.
an amazing talk, by the way.
Thanks.
Yeah, so I think like the negative membrane curvature at the junction is also necessary. So what's your take on that?
Well, I actually don't think that the membrane curvature plays such a big role, because we are talking about much, much larger size levels. So if we are looking at these vesicles here, which we hope to divide, they are relatively large. So they are in size about 10 to even 20 micrometers. And the membrane curvature, you know, from the perspective of a lipid or from the perspective of a protein, is nearly 0. So we basically, or infinity basically, so the radius. So we basically have a flat membrane for these molecules. So there must be other mechanisms. That's very important. I mean, at the end, at the scission here, I would say, yes, there we have membrane curvature plays a role, but the large way that we need to go from a round or almost round compartment that is large in size to the constriction into a stalk, this is nothing that the membrane curvature plays a large role.
Yeah, so the assumption is like whatever the force that is bringing actually into this like almost division or almost fission state is still going to be able to pull it through, that division is going to take the membrane
Yes,
curvature.
I mean,
Even at that point is not relevant.
Yes, I mean the advantage of our filaments is that they're all self-assembling and disassembling. So whatever happens at the very last instance is basically that the filaments will disassemble. But the major task that we need to find is how to make the large scale force induction.
Okay, yeah.
Fantastic. I love it. I mean, interestingly, today, basically, 5 a.m. to 9 or almost 10 a.m., Pacific time, I attended and give a talk about the minimal cell and then all the research using minimal cell. That's another synthetic cell, I would say. And then now we are talking about, very fundamental, minimal or the synthetic cell, research. And then I also hope to see more, from the Duhan stuff. Thank you so much. This is absolutely amazing.
Thank you very much.
Okay. Now, the main speaker of today with a longer introduction. Dr. Duhan Toparlak is a postdoctoral fellow at the University of Oxford, UK, as I mentioned earlier. After earning his biology bachelor degree in Turkey, Duhan was awarded a Fulbright scholarship to study chemical engineering at Rensselaer Polytechnic Institute, USA, or RPI. He completed his PhD in 19 at the University of Trento, Italy. So basically, he is all over the world in a sense, and also worked with Professor Jack Szostak at Harvard University during this time. In fact, I met Professor Szostak, a Nobel laureate, of course, in the University of Cambridge last week during the conference and had honor to present my work before his keynote talk. Following a brief postdoctoral position at Yale University, he joined Hagan Bailey's group at Oxford Chemistry in 2022 as a postdoctoral fellow supported by HFSP, I believe the Human Frontier, long-term and Marie Curie fellowships. His research focuses on single molecule chemistry and functional nanopores for catalysts, and building on his prior work in chemical origins of life, synthetic biology, artificial cells, and also in vitro evolution. Importantly, he is one of the 29 Hawaii Conference committee members who have been organizing the in-person version of this webinar series that's happening on December 9 to 12 this year, held in Hawaii, of course. And Duhan, and thank you so much for your time today, your service for the conference, and please take it away, all yours now. Thank you.
Thank you very much for this kind introduction, Tassog. I should also mention that my PhD supervisor was Sherif Mansi. Then actually most of the work that I'm going to be talking today was under his supervision. So unfortunately, I'm not yet going to disclose my postdoctoral research, but hopefully it's going to come along soon. So there we go. I cannot share.
No, I give you the permission to share, so you can click.
So I see whiteboard when I see screen. My desktop entire screen doesn't really show up actually.
Oh, really? So that means you join as the audience, not the.
Wait, I think.
No, let me. No, you
No,
join
I
as
am
the
going
audience.
to quit right now and then just join because I had some security issues. Sorry for the
Oh,
hiccup.
I see. Okay.
Okay.
So we just wait in that case. So just while waiting, I just wanted to ask, because you probably need to leave earlier. So I just curious what would be next, because your technology has huge potential to advance our science later, but simply just short answer to that question, can you?
Let me change.
Well, yeah, sorry. Just this last question. Yeah, I mean, we basically still have a lot to do in order to work on the question of the constriction. So I think the next part will basically be trying to involve also something like a genetic material in these cells.
Got it.
So that is the next big.
Okay, now there you go. I see your slide. And can you show us slide mode?
Yeah, I think it is moving, right?
No, can you click the slide show? This is currently
Yes, it is. I am on the slideshow right now. Is it all right now?
No,
We are
it's not slideshow.
seeing the full.
I see entire PowerPoint right now.
Yeah.
I mean, if you wish, I mean, you can use that one, but slideshow might be bigger.
So this is, I'm sharing my screen right now. How is this right now?
Now,
it works.
yes.
Okay,
Now it
cool.
works.
Yeah.
Wonderful.
Thank you very much, Tasok, for the kind introduction. Today I'm going to be talking about proposed pathway towards early evolution of life within prebiotically plausible protocells. The talk is going to comprise mainly my late PhD and a little bit early postdoctoral research. So we know approximately 4 billion years ago, the primordial Earth without life present more or less looked like something like this. Of course, this is an artist description, but life very likely emerged somewhere around here. So it's very basic that we know that life was emerged, but the problem, one of the question is like we have only one evidence for life in the entire universe. And then the essential questions become, how did it become like this, and why actually did it become like this? And this sounds like a little bit of a philosophical and existential question to some of us, perhaps, but at the same time that it is possible to do further research on this. And I would like to summarize here by saying, in my view, the origin of life question is actually a process rather than a one-off event that has happened on this planet. And the goal here is actually understand what kind of experiments can we do to further study and make sense of this the process itself. And obviously, we are not the first people to do so. We are actually quite late in this game, starting from the early 20th centuries, starting with Oparin and JBS Haldane. So people before us have thought of this question and said RNA seemed to be quite central. So therefore, RNA might have been actually one of the earliest polymers. So they coined it RNA world and nucleic acid first, or let's say propagation of information-bearing polymers. And some other people thought that metabolism plays a very key role on this view. So that's why some iron and the sulfur containing like some enzyme-like structures or through minerals surfaces some energy harvesting systems from the environment could have emerged, and therefore metabolism must have been already there, and then the cellular structure and the evolution took place. And some other people also thought of that, well, these two may not be actually enough. So there has to be some catalysts around or autocatalytic cycle type of things. So therefore they said, well, we see this like in the world right now that we are living with full of biology, the peptides and the proteins are governing. Therefore, peptides or catalysts of this sort must have been coming first. But at the same time, quite recently, some other people also thought that, wait a second, encapsulation of self and separation of other things that are out of a cell, let's say, is pretty important. Therefore, the compartments or primitive cell-like structures must have also come even actually perhaps before all of these, and then they coined the term lipid world. So the question is actually, whether to combine all of these together or study them in isolation. And when I started my PhD, I have actually set out to do some experiments to bring some of these structures together, meaning that I wanted to study protocell or lipids or some compartments, let's say, that are not still yet cells, and some RNA and some actions of these nucleic acids. And towards the end of my slides presentation, we will also come back to this slide, and I'll provide you a little bit more holistic view of what I actually understand by this. And hopefully I'll be able to convince you. And before moving into this, I would like to point out a new notion in the origin of life research that is actually already emerged, and everybody in the field is taking it seriously. That is called systems chemistry. In a nutshell, in this complicated slide that you have seen, similarities, in fact, between extant and the prebiotic chemistry. And what do I mean by this is what we have in our cells at the moment are very likely to be the consequence of some non-living chemistry or some prebiotic chemistries projections over today. But the question here is actually how did it become, obviously, and the decades of research has shown us that it's not actually that straightforward to come up with answers, but it is emerging that some key elements, such as this red circled functional groups or some intermediate chemicals, such as 2-amino-oxazole or some nitrile activated amino acids, may have been played some key role. And what do I want to give as a message before I start showing you some data and further ideas, that counterintuitive reality of nature, as if we observe today, may not fit with the sequentiality of a human logic. For example, the hypotheses or theories of RNA world, catalysis, or metabolism first world are mostly talking about some events like A led to B, C, D, and we see as biology today. But the notion of systems chemistry, at least as far as I can understand how it have presented, is actually there might be some hidden intermediates and some other chemical reactions that are no longer visible to us, but they actually led to a much more complicated network of chemistries and led to the biology of today. And I suppose that my talk is going to be talking a little bit of these intermediates. I would like to briefly give you the overview of what life may mean actually to some of us. And I don't think the strict definition of life is necessary, but it is also important to outline what we actually mean by life or lifelike. We know life operates out of equilibrium, out of thermodynamic equilibrium, and it happens within self-replicating compartments. Our cells, as Petra Schwille has said, divide. We want to reconstitute this, but these compartments also may have been present on early Earth. Therefore, that this growth and division or stable reproduction, if you want to call, is perhaps an essence of development of lifelike chemistries. We take some simple building blocks as fuel. We do something inside of this as some complicated or semi-complicated chemistry. We might call it as metabolism, and we excrete some of these, and then we just like reshuffle all of these things. And within this background idea is actually studying the chemistry of biology, either extant or prebiotic, is by constructing model systems that are just simplified enough. And the idea here is once we understand the physical chemical forces that drive to these primitive compartments growth and division, we might actually understanding what kind of this RNA chemistry or within these molecules, any form of nucleic acid that contains some information can have implications to the actual function of the protocell. And what we call this is a genotype to phenotype linkage. And here in a parenthesis that I would like to describe a definition from a canonical paper by Sostak, Bartel, and Luisi. And for those of you that who are interested in further background reading, I strongly recommend this seminal work or a hypothesis paper. We define the protocellular life cycle as self-replicating protocells that are not cells yet, that are not alive as considered, and some functional RNA that is taking place with the copying chemistry, that in other words, it self-propagates. So what we want to have is a system that basically self-governs itself by some using chemical and the physical conditions governed by the rules of what the, for example, thermodynamic stability or some kinetic disparities. And this system is going to be self-sustaining. And we call this as model systems. And these model systems are generally built with fatty acids. And fatty acids, in the next slide I'll describe to you, are actually thought to be probiotically plausible. And within these fatty acids, if you were to put some nucleic acids, or if you want to call as a very primitive genome, and you can call this as protocell, if you want to say. I'm sorry for the description, is actually formal description is coming a little bit late in this talk. But nevertheless, these protocells can actually host some nucleic acid copying chemistry that are very basic. But the problem of these protocells is actually they are dynamic, they are extremely interchanging with their environment. They can actually flip-flop within bilayers. They can actually move laterally within the same bilayer. They basically have so much dynamicity in the system that they can actually form some transient pores. They can grow by incorporating other lipids and so on. But the simple problem with these dynamic system is actually the salt that we see, that we put in our food and everywhere, is actually a prime enemy for these fatty acid-based protocells. And in fact, we think that the prebiotic earth had different compositions of salts, and that's actually not very much compatible with the current hypothesis of the fatty acids being model system. And in a nutshell, we have to use less than 100 millimolar of sodium salt and less than 2 millimolar of magnesium, and a very narrow pH range. So magnesium, I would like to highlight in this talk as well, it is important for functioning and copying of nucleic acids. And in fact, what you have, if you put the magnesium salts within fatty acid containing tubes and such that, and you basically shake them or vortex them, you're going to form some form of very nice soap. I don't recommend you washing your hands with it, but at the same time that you understand, I guess, what I'm talking about. In other words, system is going to basically reach to a dead end. It's not going to be dynamic anymore. It's going to collapse. Therefore, what we have thought is, are there any alternatives to these fatty acid protocells? And here on the left-hand side, you see all these fatty acids or prebiotic lipids and its derivatives. This glycerol group that is attached to the carboxylate group is somewhat providing some stability, but it's not enough. It is still dynamic, but again, it is collapsing in the presence of the salts. And on the right-hand side, that we observe a modern lipids that are pretty much similar to present-day lipids, and they have a double chain, that's phospholipids, and either with some other head group or not. Here that you see without the head group modification of phosphatidic acid. And these lipids are actually pretty stable. They don't have any problem of that sort, but they're not dynamic. What do I mean by this is you cannot actually use these lipids to induce growth and division events that we are going to observe in a couple of slides later. And therefore, the logical conclusion is perhaps during this green to red transition, there were some other intermediate species, such as this one with some cyclic phosphate head group, or even linear form, so that like the system reached into a much more stable and controllable way. And The idea is actually coming from partially from Ram Krishnamurthy's laboratory at Scripps. We collaborated with him during my PhD. Then we were actually coining the term whether these cyclophospholipid can be alternative to these fatty acids. And they kindly devised a pragmatical plausible chemistry, meaning that they have started with the short chain nonanic acid, carboxylic acid here, with the glycerol in the presence of the glycerol and some diamide phosphate, which is a very efficient phosphorylating agent under certain prebiotically plausible conditions. You can have a good conversion yields to these type of lipids, and they were shown to have some vesicle forming abilities. And we have actually asked the question, number one, do these phospholipids are actually cyclophospholipids form stable protocells, meaning that in a hypothetical scenario, we can start with a simple system of carboxylic acids that are collapsing, but then at the same time, somewhat along this gradient of changing pH, sodium or magnesium salts, as vesicles over time become more stable. And the short answer to your question is yes, we have shown this along a very broad range of pH. I'm not showing you that to skip some of the messages here, but from pH 5 to pH 10, the one that I have tested, they were pretty stable. But one of the important factors here is that was attractive, the near seawater levels of sodium ions that are present along with some magnesium ions rescue the protocells from total destruction. Here on the very much right-hand side, you see a fatty acid vesicle that is basically formed into these soap-like structures that I was telling you. But at the same time, under same conditions, identically, cyclophospholipids are thriving. They have really nice round and like very dynamic tubular-like structures. And the conditions basically were up to 25 millimolar of magnesium in the presence of 400 millimolar of sodium, that you were basically able to screen all the surface charges of this phosphate head group, which is negatively charged, and very likely that the remaining magnesium ions have no longer possibility to interact and they make the system collapse. This was an interesting finding, but it was not enough to further expand our research. Then we started to ask the questions, and the second question, what kind of scenarios enable sustainable growth and division of these cyclophospholipid-like vesicles? But then we wanted to understand whether we can actually achieve some iterative growth and division cycles. In a minute that I'll also tell you that we are nowhere near the first one to propose these, but at the same time, we are trying to make the system a bit more plausible. And along my studies that I was characterizing, I actually said that, wait a second, maybe actually we don't even need to actually understand some of the challenges with the cyclophospholipids, but just using this even more simpler system as fatty acids. Here in this slide that you basically see vesicles fed by vesicles. Let me tell you, here is a brief intro to the topic here as well. Pioneered by Ting Zhu and Jack Shostak in 2009, the fatty acid vesicles were shown to grow and divide when you add some micelle structure. The micelles are basically non-vesicular aggregates of fatty acids, let's say, that are not necessarily just fatty acids. It can be any form of amphiphilic molecule that is no longer in a bilayer form, but just collapsed onto itself with the hydrophobic core. And these structures are somewhat unstable, and they want to be even more in a thermodynamically stable phase, which is in this case, if you reduce down the pH, that is in the bilayer. And if you increase the amount of these structures, the vesicles can be fed into the bilayer, and these basically create some surface to volume imbalances, perhaps expanded by the inflow or outflow of water molecules in the presence of some water impermeable additional solids, and form these filamentous structures. And if you shake them very briefly, they basically divide very nicely. So you can have lots of daughter vesicles. And lots of different modalities of this sort of growth and division cycles have been shown. For example, you can have different solutes, osmotic pressure, or different phospholipids. Again, not prebiotically plausible, but somewhat reaching to a different line of chemistry towards complexity, or some hydrophobicity or even concentration-driven growth has happened. But in all of these scenarios, there is an extra element. In other words, that we need to disturb the system, and the system always going towards the equilibrium. And this is not surprising, but we wanted to achieve even further plausibility within the system. In other words, we wanted to understand whether we can feed the vesicles with vesicles in all the crude way possible, without myself, without any final control, etc. In collaboration with Anna Wang from University of New South Wales, we have basically understood that these vesicles can be, again, fed with vesicles if you have different lengths of a fatty acid chain on the hydrophobic end. In other words, the diversity of the vesicles might actually have led to the growth and division, and all of these systems can take place in an iterative cycle at the population level. And the difference is just only a few hydrocarbon, few carbon change. So in other words, that we usually fed with the 18 carbon chain with the 14 carbon chain, or vice versa, or 12 carbon chain with the 8 carbon chain. So only 4 carbon will make a big difference here. And as I told you already, give you the idea, the long chain vesicles, when we have them in isolation and we feed them with the short chain vesicles, the system basically accepted all the short chain vesicles and the monomers in them, and basically they formed the nice filaments and protrusions, and eventually through mechanical force they have driven towards the division. This was not much surprising, but what was more surprising to me during this characterization was there was a coupling of growth, automatic, almost like unintentional coupling of growth and division of these sort of vesicles when we have put the short chain vesicles and fed with the long chain vesicles. And here is the video of how the system, within a couple of seconds of addition of the lipid, that you basically develop a sort of a budding of event from perhaps an outer shell, very likely to be some kinetic disparities. And along the way that you see, even the other further daughter vesicles keep growing by themselves, and they basically form the filamentous structures. And these filamentous structures, if you isolate them somehow within the system and then reshuffle with the different lipid composition, they're also going to grow and divide. And this system, in principle, can take place indefinitely in all iterative cycles. And the reason why it has worked is simply because we had the difference between the kinetic disparities, and I'm going to talk to you a little bit about it later. But I have to tell you that this system works not only with prebiotically unreasonable lipids such as with 18 carbon chain, but also with the C8 vesicles, that is octanoic acid, or lauric acid with the C12 vesicles. And they basically, again, in the most crude way, they take place. You don't have to isolate so much these vesicles, so you just make them. You just need to put a little bit like a heat in the system, because some of these lipids are quite solid at room temperature. But there is not a problem with that in terms of prebiotic earth, because we know, give or take, we estimate the temperatures were highly, a little bit more elevated, not so high, but a little bit more elevated on prebiotic earth. And why this happened is basically almost threefold in almost every cases. The lipid flip-flop rate is basically from inner bilayer to the outer bilayer is basically the one of the critical factors. It's just tiny bit of a difference of this 4 carbon difference between 18 to 14, let's say, is able to indicate the net accumulation of one form of lipid on the outer shell, because before it reaches to the equilibrium, there is no more chance of this outer shell to hold any more bilayers, and it essentially leads to some form of membrane restructuring, and this net accumulation within a couple of seconds can make you actually reach to these thermodynamically more stable structures, and therefore some form of division that you see. So then the third question was, do these cyclophospholipid protocells that we have shown were prebiotically plausible, and they can endure some high concentrations of salt, can also divide and grow in the presence of magnesium, and or they actually potentially couple the RNA copying chemistry within themselves? Briefly, this is the last work that we have done, the published late in last year, December. So these cyclophospholipid-rich vesicles, then when they were fed with fatty acid vesicles, well, not much to your surprise by this time of this talk, they grew and divided both with and without magnesium. So there were no problem, nothing has been hindered by the presence of magnesium. The protocells have grown and divided a little bit better in the absence of magnesium, but perhaps it is also important to note that it is compatible, these concentrations of magnesium, that is a citric acid chelated, that I'm going to be coming to that in a couple of minutes. It is actually probiotically reasonable system that perhaps can achieve some form of RNA chemistry with this concentrations of magnesium. This is basically pushing the system into its limits in a way, but at the same time, as you can see in all these slides, that the tiny bit of these dots are all individual vesicles that starting from, for example, like a handful of them, after 30 minutes or so, that you're reaching to the thousands of them. So this system is quite robust to reach this either magnesium or magnesium independent or independent division. And again, to give you a little bit more understanding of what exactly causes this growth and division observations, is simply put, instead of putting the fatty acids into the vesicles with the different hydrophobic chain length, now that we are working with the cyclophospholipids, and what we have characterized is, due to the hydration of the head group, which is very much water-loving group, even more so than the carboxylic acid, cyclophospholipids, simply put, they enter into the bilayers much faster than the fatty acid counterparts. And eventually this basically, again, as you might guess, is going to lead to net accumulation of the lipids on the outer shell. And again, indeed, furthermore, the cyclophospholipids, even to not too much surprise, because they have negative charge at the physiological pH or near physiological alkaline pH, 8, for example, or 7.5, they also flipped towards to the inner leaflet slower than, again, the fatty acid counterparts. And this basically two net effects of first jumping into the solution quick and an inability to flip-flop from outer to the inner part of the membrane essentially led to some form of kinetic disparities and the net accumulation of the cyclophospholipids at the outer layer. And then eventually everything basically wants to stay in the nice rounded shape, because that's, again, thermodynamic, the most favorable phase for these lipids. And at the end of the day, we have observed this division, as you have seen in the video. And furthermore, as I was commenting before Petra's talk, we believe basically the positive or negative or positive presence, in this case, curvature of these structures right at the time of the division might have been actually somewhat important to trigger some of these effects. Again, in the macro scale of the things, obviously, these are not so relevant, but just right at that moment of division, some of these effects might have been present and further facilitate or ease the system. And in this case, most of the time, with the biological systems that we are actually talking about net negative charge or net negative curvature in the system, especially in the junction. But at the same time, well, we are not necessarily dealing with so much of a biological system. So there might have been some other physical modes that are leading, because we know the geometry of these lipids, because of that bulky head group, is definitely creating some structural differences, because we have constantly observed the profound differences between the carboxylic acids and the cyclophospholipids. And again, moving towards, so what is actually useful or beneficial for this type of growing and dividing, and there's just some details on the lipids. So you can ask this question, well, are they stable enough to hold some nucleic acids or RNA or DNA, etc.? And the short answer to you, actually the best answer, even with the prebiotically plausible versions of these lipids, which is basically some 10 carbon or the 12 carbon versions of these, the answer is yes. They can grow and divide very nicely. We have also shown the short chain lipids, fatty acid or the cyclophospholipids can actually grow and divide, and they can actually also retain the whatever is encapsulated within them. That's very good news, because if we do have this during growth and division, which we can, again, by introducing different types of lipids, we can achieve the iteration of these growth and division cycles. And the idea is, if they also retain the RNA molecules within them and some chemistry, if everything is allowing to do so, some, for example, copying of a very primitive genome, we might actually have a very strong model or protocellular life cycle within our test tube. And in collaboration with JaxoStack, that we have basically devised their non-enzymatic RNA copying system. That is basically here, as you see on the left-hand side, 2-aminoimidazole activated, just the GMP is a very simple G monomer is going to make a hydrogen bond with the C on the template structure, and in the presence of some magnesium and the street molecule. This basically is a chelating agent that is not dangerous to any form of membrane or something, as we have shown before, and it actually also allows this RNA copying chemistry to take place, and as we have shown here, within the vesicles, either with the cyclophospholipid deficient vesicles or with the cyclophospholipid rich vesicles. In both directions, the system actually can allow the passaging of these monomers together with the magnesium ions within, and it basically allows the copying chemistry. But there is one big problem with this system, and that is the rate of copying chemistry is nowhere near as fast as the rate of growth and division of these systems. In other words, if you want the system to be somewhat prebiotically functioning and like leading towards some Darwinian like evolutionary system, if you have some functional RNA encoding some whatever the chemistry that you want to work with it, actually the copying of these RNA molecule is nowhere near as efficient, and that's actually a big problem and further subject to the research. But again, in principle, within this study that we have shown, the RNA chemistry can take place, particularly the copying chemistry can take place without the presence of enzymes. So then we have a system, in summary, that has some lipid imbalances, lipid differences. You can basically feed this, and they're pretty much prebiotically reasonable. You can basically feed the system with either form of lipid. You can encapsulate RNA. You can copy this RNA, if you will, and you basically form some daughter vesicles. So then the immediate question is, what is next? So we have this somewhat model that works, but we need to optimize and further show something useful with it. And before that, I reach to my conclusion slides, that I would like to give a brief overview of what do we mean by these systems, and what do we mean, like what do we want to do with this exactly. So if we talk about this time course of this emergence or origin or let's say early evolution of life, like systems, that we can actually say the system overall, whatever the protocell or nucleic acid or some lipidic systems, actually reach to a further complexity by storing even more energy within their bonds and so on. These prebiotic operations basically reach to somewhat a transient intermediates that are self-assembled, and they can actually behave some of these lifelike systems and some interesting or dynamic chemical reactions, as I have actually shown you, either in the micro scale or in the macro scale, or if you want to say, in the supramolecular scale. And within this energy or let's say the complexity curve, that you have basically somewhat a stable system that is far from equilibrium, that reach to a somewhat a sweet point in energetic system, and it basically reaches somewhat metastable point. And within this metastable point, one of the leading goals right now is to achieve such form of metastable systems happening and basically reaching to somewhat cyclic events of, for example, growth and the division or reassembly of the systems and the redistribution of the system. If you want to define these and if you want to make these prebiotic, this form of life cycles or this form of a dynamic chemistries might actually eventually lead into more and more gradually increasing lifelike chemistry, and perhaps at some point some Darwinian factors such as natural selection or some selection pressure happening through these fine nuances of the chemistries, as I have shown you, for example, these 4 carbon differences or some head group differences, or if you want to say some more biologically looking alike structures or ability to acquire metabolism sustained somehow in this cyclic version. And if you integrate some of these parts into growth or division or some nucleic acid structure, we can keep maintaining this system out of equilibrium. And perhaps at some point that we are going to draw a line and say, like, well, after this point, it's kind of life, and before that, it wasn't. So there is no line, in fact, but it's all gray zone. And as I said in the previously, it is actually a process, and we would like to understand. Coming to my conclusions, I'm sorry if I have gone over time a little bit. In these 4 pillars of origin of life ideas: RNA world, metabolism or iron sulfur world, peptide world, and lipid world, the modality of understanding of them is actually changing. We are observing with more and more interesting data. Ideas have been tested, and we go back and refine our hypothesis, and we restructure our hypothesis, because this is the essence of doing science, and this is the correct way to do so, not to sticking with our ideas. And within this mode, a systems chemistry structure, a more holistic view of all these elements are coming together and bringing perhaps the pieces together. Everybody has perhaps a different understanding of what systems chemistry is, but I would like to briefly summarize what actually current understanding in terms of all these 4 pillars. On the left-hand side, that you see the RNA is actually sort of present along with amino acids. So that's why you can coin the term RNA and the peptide world, because the chemistry that lead to the RNA's prebiotically plausible RNA synthesis might have been actually also quite plausible with the peptides. Therefore, we conserve the information here, and that actually lead to some functionalities and some interesting chemistry that no longer observable to us, but have happened with that we know that we call activation chemistry, let's say, or protometabolism, and that actually leads towards complexity. Some of the remnants of this, for example, in the form of thioesters, can be present in our bodies right now. But again, we actually don't know exactly how the system is going towards. And within this activation chemistry, some autocatalytic sets or some self-sustaining systems could have been actually present to conserve these chemical reactivities and finally if some lipids and some phase separating conservates, liquid-liquid phase separation, can actually form some functional compartments together with all these like three other systems here together that we're talking, perhaps an emergence of cells will take place. And now that we see these within this grand scheme, we actually with prebiotic reasonable model system we have that we can actually probe the conditions within this overall holistic view. Some Darwinian evolution might have been taking place, some different forms of RNA copying and the RNA sequence space, and many other interesting questions. And in my last slide, I would like to give you the message here. If I haven't convinced you already, I believe for the emergence of Darwinian evolution, a little makes sense without protocells. And this is basically inspired by the idea of saying nothing in biology makes sense except in the light of evolution. So for evolution, we need compartments. And thank you very much for listening to me. I'm happy to welcome questions, and I would like to also acknowledge my past supervisors and my current supervisor and my current funding agencies. Thank you very much.
So absolutely amazing talk. So, also very interesting because just before this seminar, chairing, I gave a talk for the minimal cell workshop, hosted by the John Glass at JCBI. And I talking about, many interesting overlapping, concept. And I talking about the 7 governing principle in biology, and that I published recently. And then, of course, the main thing I kind of talk about is the evolution, of course, and also, the robustness or the entropy increase outside, but internally entropy decrease as an unequilibrium system you're talking about. So absolutely beautiful. Petra stay, more than 1 hour, but she need to leave. So
Yeah.
before I asking my question, I want to ask her question. She left a question in the chat. My question would be, what step would be most pleasurable energy dissipating reaction for these interesting dynamic lipid and co-acerbate system to divide? That's interesting.
That's actually $1 million question. I mean, I appreciate that you ask this because this is also the head scratching to me as well. And actually, there are a lot of steps, and we also need to be really careful about how we define actually the energy dissipation. So these are the like buzzwords terms, and we can actually use them quite freely. It's like as a can of Coke and like opening it like a pop soda. But the point is we need to be very careful about like what are these systems actually mean. So I would say again like a acquisition of a different head group overall, because the system, like in a protocellular system, like a membrane system, let's say, has to host some amphiphile at some point, and you need to differentiate it. And the differentiability is either going to come from the head group or coming from the tail group. So if you have the tail, that you basically reach to a kinetical trap system. So the more that you actually play with the head group, is more likely that you're going to introduce a bit of a difference in the system. Again, as I've shown in the presentation, that might actually lead to unexpected, interesting division like characteristics. Yeah, but it has to also be coupled to some form of a synthesis chemistry or some dynamic chemistry that is going to be present either on the outside or within the protocells. Yeah.
Okay, wonderful. Before I ask my serious, also very challenging questions. So you mentioned one paper authored by Jack Shostak and Luisi. You're talking about Ben Luisi, from the University of Cambridge, right?
No, it was Pierluigi Luisi, the Italian
The other
scientist
person,
from
okay.
ETH Zurich.
Okay, I tried to find that on my Kudun. Okay. So my real question is,
I can tell you the title. Title is the
sure.
Synthesizing Life, 2001 Nature.
Okay, so you have only the name of the author, but I couldn't find that. Okay, wonderful. So my real question is also very challenging or philosophical in a sense. Question is, I love movies. One of my favorite movies is The Matrix. You watch that one, no?
Yeah.
Okay, so Matrix, basically the word repeat, I believe 7 different times. And then my question is, because you're talking about evolution, so if we repeat the world or the universe 7 different times, are you expecting same lipid bilayers with the same molecule, or we are talking about different molecule for the,
Not at all. Chemical
I don't know.
reaction. The governing physical or let's say thermodynamic principles are going to be the same, but let's say like not the same, but going to be probably similar. Exclusion of water around so that like molecular interaction is going to take place, so that they
Okay.
want to stay in the stable form. But lots of chemicals, different chemicals can do that, and they do that all the time. Different lipids exist in archaea, bacteria, prokaryotes, even eukaryotes. There's so many different versions of them. It's not going to be the same lipid, I would nearly confidently guess. Can be very exotic, block copolymer type of things, not necessarily phospholipids or any form of water-loving and water-hating parts, basically,
Okay.
from basic biochemistry idea, let's say, that can actually bring these things together, that can form some hollow shape that can isolate themselves from the inner materials, from the outside material. That's it.
So that means, but my question is much more broader. So that means 7 different experiment or 7 different universe. We are talking about Earth. You are talking about we may have 7 different type of lipid or 7 different type of molecule, not just lipid, but phospholipid, some different, complete different type of the molecule encapsulating the cell. That's my question.
Yeah, a lot different. Either way is different.
So that means this is
Either
your
way.
view, or this is the general view from your community?
From assuming that prebiotic plausibility is not the question, I would say people would guess that's the case, yes, because you can have like cationic lipids, for example, with some amine head group or is even actually like very much stable up to crazy concentrations, like almost like molar concentrations of magnesium and stuff like that, some of the lipids. But that's not like what we observe in our bodies, and we cannot actually deduce some like linear pathway towards our modern life, what evolution actually brought us. So yeah, lipids exist all around. If you want to say like in the 7 universe or within our universe or within our galaxy or in another galaxy or planet, I am sure that there are interesting, if the prebiotically plausible chemistries are similar, fine, perhaps we are going to reach to some form of variations of what we have. But let's say we don't know, like for example, like icy moons of like Jupiter, Saturn, etc., or some other different like non-water containing like hydrocarbon like lakes exist in different planets, right? On that case, if you want to say phase separation, if you want to call the lipid, what is lipid? So you're not going to exclude water in this case. You're going to actually exclude your hydrophobic molecule.
Sure.
So then you have the lipid function by doing like inverse mode of like what we observe here. Like
Sure.
their hollow is in that case, if there's a microbial or protocellular life, whatever you want to call, is gonna be in like a hydrophobic or alkaline environment.
Got it. So you probably expect my next question, what my next question is. That means if we do the same experiment in parallel, starting with Big Bang, you believe in alien.
So here's the thing. I think the microbial life, my students also ask this question all the time. Are we alone? What's going on? Like how is it? Like, I mean, I wake up, I always give them this example. One day I wake up and I say like, oh yeah, there has to be so easy. Chemistry is all around. Like it's all the same molecules. It's so like everything falling into place. So there has to be some other places in the universe. And another day I was like, yeah, it's actually, you know what? They're kind of difficult to do that, this type of stuff. So here's the deal. So yeah, but here's the deal. There is always some level of scientific understanding within this. And we can actually, if you think about it, we are the universe that who makes sense of ourselves, and we can actually understand what's going on. And we can actually draw lots of abstract analogies. And like, what are these molecules that we're dealing with that we know that actually it belongs to us? And at that point, what happens is like, oh, wait a second. So it doesn't really have to be like 4 billion years, 5 billion, whatever. If we have the proper conditions in a given position in the universe, it actually kicks off like pretty quickly. Like, for example, there was a recent paper we have actually reviewed, like they said, like one of the earliest microbial life, like the progenate of current microbes, might actually existed like somewhat 4 billion years
Okay.
earlier. So that basically tells us, because now that we have been, until that paper, claims of that paper, we were saying 3.8, 3.6 billion. But like, wait a second. So that's basically Earth formed like 4.54 billion years ago. And like, we know that it's so like hot, there's like a molten lava, etc. everywhere. There is this moon forming collision, everything. After that immediate cooling down, it's like, we think that this is 4.2 is like 200 million years that you're telling me to lead to the complex microbial life is actually pretty amazing to me. Like just that is basically telling how much chemistry had to be poured in to reach to that complexity, is basically telling me that life is supposed to be perhaps everywhere in the universe. But then the question is like, where the hell are these like rest of the microbes that we cannot see? So yeah, short answer to you, maybe yes, maybe
Sure.
not.
That the reason I'm asking that question is in my article, I call it 7 governing principles in biology. I'm talking about multiple different things, including central dogma, evolution, etc. And I always wonder why we have the 4 bases for the DNA and only 20 amino acid. If you include the whatever extra amino acid that it could be up to 22 or whatever, why not the 18 or 19 or 5 bases, etc. And then I do believe in somewhere in the universe, there are some living system that containing 6 bases rather than 4, etc. And I just wonder, but this is a fabulous, but still we don't know, the answer because we have never have evidence,
Yeah,
clear
these are
evidence.
extremely nice questions, and I can tell you, active researchers are constantly asking these,
Of course,
why these like 20, why these like 4, and I think you can actually bring down the question further. We know other nucleobases exist, but we know also other sugars exist. So
right,
why RNA
right. That's
specific?
what I'm talking about. Why
I mean,
deoxy? Why that sugar, not other sugar, right? So
if you go ask John Sutherland and Jack Szostak or Matthew Powner, so these are organic chemists and like the pioneers in the field, they're probably going to tell you, well, synthetic pathways lead to that. But if you ask me, they're also doing their research. Jack Szostak will probably say, well, copying chemistry must have been like even more efficient with RNA and stuff. But we don't know exactly what copying chemistry was actually present at all. So that also has to be understood. Maybe I don't know, maybe there was some other chemistry somehow copied like 4 carbon chain, like nucleic acid, for example, with the
sure.
ACTG, exactly like all these
Yeah,
nucleobases, but like the sugar or even actually phosphate head group.
yeah,
Say, sorry, not head group, the backbone itself. So
yeah,
it doesn't have to be phosphate. It can be like
yeah,
alkylated, methylated. I mean, there's so many, so many things.
yeah.
Yeah.
Yeah, I have honor to talk with him during his talk and after his talk or before his talk actually. in Cambridge. And then I asked some question, but not all, because I need to leave the conference just the first day of the conference for the second, conference in Germany, unfortunately. But I remember. Also, I want to invite him to this seminar series, but I haven't got his response yet, unfortunately. Also to Hawaii, but hopefully you help me to recruit him to the conference and the seminar series. Okay, so we need to be mindful. We are way over, so 11:20
Okay.
Central Time. So I need to close. Let me double check whether we have the question from audience. We don't have today. So we have the
Oh, I just like responded to
oh
you
the
give
chat. Okay, the title. Okay,
title, yeah.
so let me close and then we probably chat more
Mhm.
after stop recording. Okay, so thank you all for joining and staying today. We'll meet again on October 3rd, already October. Thursday, the same time, the same Zoom link. We'll have Professor Elisa Franco at UCLA, and then Professor Justin English at the University of Utah. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. I will stop recording in a second. Thank you again. Just 1 second. Stop.